Alright, so we're going to look at Chapter 5 in our micro textbook, and we're going to do this text in two separate sections. So this would be Part A of Chapter 5. Starts on page 124 of the textbook. And so in this chapter we're going to focus on eukaryotes. If you recall, in the previous chapter we looked at prokaryotes and bacteria. We talked about just some basic characteristics of them, and then we looked at structures from the outside in. And so then in this chapter, we're going to focus on those that do have a true nucleus. So when we look at basic eukaryotic cell structure, by the way, this is going to include our protozoans, our fungi, our algae, and our helminths. And so this is a basic eukaryotic cell, and this should look very familiar to what we talk about in biology. And so you see the nucleus, and then you see all these other membrane organelles that are found throughout the cytoplasm of a eukaryote. So if we talk about the basic plan of a eukaryote, we have some very consistent structures. So typically a eukaryotic cell is going to have one or several external structures that we'll talk about a little bit later. So appendages like flagella, cilia, glycocalyx. Eukaryotic cells will have a boundary. So that could be a cell wall and or cell membrane. And then of course organelles. So if we look a little more carefully, external structures, we just said there's the boundary, the cell wall, the cell membrane, and then all of these organelles and their components. And so in this chapter and in this section specifically, we're going to look at these structures. Now all of this should sound redundant because this was everything that you looked at in biology in that Unit 2 when we do cells and cell parts. So we start with external structures. We look at appendages like flagella. Okay. Now, if you'll recall, prokaryotes can also have flagella. Their arrangement is a little bit differently, but the specific difference has to do with the way they move. Remember, flagella is a motility appendage. In prokaryotes, if you recall, it spins 360 degrees, so it moves like a propeller. However, in eukaryotes, the flagella whips back and forth, and so as it whips back and forth, it propels the cell through the environment that it's found in. So it's a big difference that we see as far as movement in prokaryote and eukaryote flagella. Now, a new appendage that we see in eukaryotes that we don't see in prokaryotes is cilia. Now, a lot of people confuse these with fimbriae. Their structure is a little bit different, and again, fimbriae found in prokaryotes, cilia found in eukaryotes. Same function, it's a locomotor appendage, so it's for motility, and essentially, structurally, it's the same as flagella. Biggest difference is the length. So where you look at flagella being this really long whip-like tail, cilia are going to be much more shorter, hair-like structures. But again, same function as in motility. These can also be used to kind of sweep food in. So if you look at this particular protozoan, you can see where it will sweep food into what they call the oral groove of the gullet, move it down towards where it can actually be processed. And then of course it can also be used for filtering. So you have the flagella, you have cilia. Another external structure that you can see on eukaryotes is the glycocalyx. Now, again, we talked about this with prokaryotes. So it's that sugar coating, lots of polysaccharides. It also contains some proteins. And again, same function that you see here. So the sugar coating can be used for protection, can be used for adherence, but then there's a third function here that's added that you don't hear a lot about with prokaryotes, and it's their ability to receive signals. And so the glycocalyx can help in that whole communication. So to talk a little bit about difference between the glycocalyx with fungi as well as with protozoans, you'll see a little bit of what's different below them. So with fungi, it would be a cell wall below the glycocalyx. With protozoans, they don't have a cell wall, so underneath it, you'd find the cell membrane. So if we go deeper and we look at the cell wall, a couple things that we'll note here. Now, cell walls are not found on all eukaryotes. Typically, you'll remember, all prokaryotes have a cell wall, except for that one group at the end, the mycoplasms. When we look at eukaryotes, though, there's only certain types of eukaryotes that have cell walls. So fungi being one of them, they have a cell wall. The difference we see here, though, is that the cell wall of a fungus, it's not made of peptidoglycan. It's typically made of chitin. You could see some cellulose in there that's similar to like plant material, but the main thing you're going to see is chitin. Very strong polysaccharide, which is one of the reasons why fungal infections are so hard to get rid of. For algae, they also typically have a cell wall, but the chemical composition, it varies. So you can see something similar to like cellulose, pectin. It also mentions some calcium carbonate in there. So depending on what type of algae it is, it could have a different composition to the cell wall. As far as underneath the cell wall, you would find the cell membrane for fungi, for algae. If you look at other eukaryotes, the cell membrane is the outermost layer because they don't have a cell wall. Now, we learn in biology the cell membrane is a phospholipid bilayer. Here's that fluid mosaic model that you look at in biology. It's embedded with proteins and it is selectively permeable. So same concept there. Biggest difference that you'll see with the cell membrane of a eukaryote versus the cell membrane of a prokaryote is, remember, cell membrane for a prokaryote is also where a lot of their energy reactions happen. Because remember, they don't have all those organelles to do those different reactions. Eukaryotes do have all those organelles. So typically you don't see a lot of energy reactions happening here because they happen inside of all these other structures. Okay, now this is a table that's found in your book. You can go back later on and look at it if you choose to do so. But we're just going to take a few minutes and go through these other internal structures. Now I'm going to go quickly just because you did this in biology. So we start with the nucleus. Of course, if they are a eukaryote, the nucleus should be a very prominent feature. It's typically located towards the center of the cell. It's called the command center of the cell because it contains the genetic material that controls cell processes. It's surrounded by a nuclear envelope. It has chromosomes, and then in the center, remember, it has a nucleolus that helps to make ribosomes. Remember, to divide the nucleus to make new cells, you do the process of mitosis. Remember we talked about that, the PMAT, prophase, metaphase, anaphase, and telophase. Next to the nucleus, you typically have the ER, endoplasmic reticulum. You'll recall from biology that it's a network of tubing, and it's divided into two sections. You have the rough ER and the smooth ER. You'll recall the rough ER is rough due to ribosomes on it, and so because there's ribosomes on it, remember the rough ER is involved in protein synthesis. The smooth ER is smooth because it lacks ribosomes. Therefore, it's not involved in protein synthesis. Instead, remember, it's involved in lipid synthesis and storage. Typically, when we finish talking about the ER, we talk about the Golgi because these all kind of work together. Remember, so you've got your nucleus that's making ribosomes. They leave through pores and attach to the rough ER, make proteins. Same time out here with the smooth ER, you would make the lipids. And then vesicles take those materials to the Golgi where they are modified, stored, packaged, shipped, etc., etc. So this would be your Golgi apparatus. A lot of times the Golgi apparatus is known as the post office because of its modifying, packaging, and shipping capabilities. So you kind of see how these things work together, like we just said. The nucleus, inside there you make the ribosomes, those go to the rough ER, proteins go to the Golgi, from there vesicles ship them or secrete them to other locations. Mitochondria is possibly the most popular part of a cell. We all learn it's the powerhouse of the cell. But I always tell my students to make sure when you say that it's the powerhouse of the cell that you're understanding its function. The reason it's called the powerhouse is its ability to produce energy. And we learn in biology that energy in our cells is in the form of something called ATP, or adenosine triphosphate. Chloroplast. Not found in all cells, only found in algae cells and plant cells, but they have the ability to take in sunlight and use it to convert chemical energy, sunlight into chemical or food stored energy. So they are photosynthetic because of their chloroplast. And then we also mention here the ribosomes. So we've already mentioned ribosomes are heavily associated with protein synthesis, made in the nucleolus, and then either found attached to the rough ER, or sometimes they can be found free-floating out in the cell. Now, be mindful, Prokaryotes also have ribosomes. The difference we see here is the size. So in a eukaryote, the ribosomes would be much larger than they were in a prokaryote. And then to finish out cell structures, they end the section looking at the cytoskeleton. So remember, the cytoskeleton basically does the same thing for a cell that our bony skeleton does for our body. So you've got a framework of proteins and fibers. Those kind of interlock, and they give you shape, they give you support. They can also be used in movement. So the last thing I wanted to leave you with in this section answers objective number 3. So objective number 3 says, okay, now that we've talked about all the parts to prokaryotes and eukaryotes, let's compare. Let's see who has what or who's missing what. So this slide, as well as this one, will carry you through kind of differentiating between those three. So I won't take time to go through each one of these, but I would just say it might not be a bad idea to take that objective number 3 and divide it into 3 columns. So you have a column for prokaryotes, a column for eukaryotes, and then viruses are out here just to remind you that we do talk about them in this class, but they're not going to have all those cell parts because, again, they're not made of cells. But it might not be a bad idea to divide it into the 2 columns and then just to quickly go through and list the structures that can be found, give a plus where they are found, and a minus where they aren't. So like, for example, for nucleic acids like DNA and RNA, they're found in both prokaryotes and eukaryotes. But as far as a nucleus to contain them, only found in a eukaryote. So again, really good charts. If you want to copy the charts all together, that's fine, or you could just kind of find a form so that you'd know the differences. So obviously, if that is an objective, then there will be some test questions that are wanting you to make sure you know what prokaryotes have and don't have, and then as well as what eukaryotes have and don't have. And that's the end of this section.